Okay, I'm going to talk you through how to write a practical report and give you some tips and ideas on how you know if you're doing it right. Okay, first of all, what's the purpose of a practical report? The purpose is that as scientists we need to show our thinking. Another scientist should be able to repeat all of our experiment and get exactly the same result. So if I say I'm folding the page vertically, where on the page am I folding it? Am I folding it directly in half? Am I folding it in thirds? Which way is up? You know, if I don't give exact instructions in my method of how I did it, then someone else is going to get a different result. So the other thing that we want to do is clearly explain the theory that we already know before we start the experiment, so that we can show that through the experiment our understanding has changed. The first section of a practical report is the aim, and this is probably the shortest section of a practical report. First of all, we want to know, what do we hope to find out, or what are we investigating? So once we know what we're talking about, the rest is actually quite easy. All we need to do is start to discuss what the theory is that we're investigating rather than what we're doing. And we usually start with a sentence stem like to discover, to investigate, or to find out. So here's an example. I'm going to get some plants. I'm going to put one in the light and one in the dark. That's what I'm doing, but what am I going to write as my aim? Well, to investigate the effect of light on plant growth, because that's really what I'm doing, isn't it? I'm not seeing what happens when I put one in the dark and one in the light. I'm trying to work out what's happening to the plant growth when I change the amount of light the plant gets. Another way I could say it is just to find out if the plants grow taller with more light. And now I've said something that's quite measurable. Okay, background information is the second bit. Remember, you can pause this presentation anytime you want. So if you're trying to write the aim, why not pause it, write down the aim and then start again. So background information. This is where you get to say what you already know about this topic. It's really discussing the theory behind it all. So if we go back to that plant example, well, how do plants grow? What do they need? Why is light important? So you'd probably be talking about things like photosynthesis and how plants make energy from the sun. You'd acknowledge any scientists that had already studied in this area. So in your reading, you'd be explaining what they've already taught you. You might need to do some research for this section, or you might need to go back to your workbook and have a look at what the teacher had shown you. Sometimes you'll get some guiding questions to help you answer it thoroughly, and that's a bonus. But we don't answer it like question 1, question 2, question 3. We have to write it in like a short essay. So we only use the questions as a guide. Here's an example of some guided questions in a background information of a practical report about microscopes. So you can see here there's 4 questions, but we don't want to write it as question 1, question 2, question 3. So we need to be able to write these answers in paragraphs. So the first thing is, what are the main points? Well, when I look at these sentences, the main points are, what are the key features of cells? What are microscopes? And what's their history? Have microscopes always looked the same? So now I'm ready to write my paragraphs. I need to start thinking that a paragraph always has a topic sentence. So that's usually where you turn the question around and you now make it a sentence. And then you need a couple of sentences with some evidence in it to support the point you made in the topic sentence, and then a summary that concludes it all. The purpose of this text is to inform your reader of the background and the theory. So it's a factual text. It doesn't involve emotive or persuasive language. It's simply passing on facts. And because it's an informative report, at times you'll be talking about the past, sometimes you'll be talking about the present, and sometimes you'll be talking about always, which is again present tense. So here's a sample response to a simple guide in background information. What are the key features of a tree frog? So first steps reflect the question in your topic sentence. So I might write, the key features of the tree frog are its green slimy skin and its sticky feet. So I've answered the question, but now I want to give it a little bit more detail. So I'm going to expand each point I made in the topic sentence with a couple more sentences. So here, the green slimy skin is perfectly suited to the tropical amphibian, as it's often moist from humidity and living in ponds. The sticky feet allow the tree frog to cling to the side of a tree, even though the tree is so big and the frog's feet are so small. And then, of course, I've got to summarise this paragraph, otherwise I'm just leaving the reader hanging. It's these key features that allow the tree frog to live comfortably in its natural habitat of the tropical rainforest. Did you see how I went back to the first sentence and answered the question again? Pretty tricky. Okay, safety. Again, if you need to pause the video, go for it. In this section, we're looking at the instructions of the method and the materials that we're using, and we're really conducting a risk assessment. We're thinking to ourselves, well, what could hurt you or others if care is not taken? And if I'm using chemicals or sharp things, how should I dispose of them so that I keep everyone safe, including the environment? You don't want to be pouring chemicals down the sink if they're not safe for the fish and for plants. You must include the danger, the prevention of harm, and the outcome if safety procedures are not followed. I'll show you how to do that in just a moment, but first, there's 3 types of hazards we commonly come across in the lab: biological hazards, chemical hazards, and physical hazards. Biological things are all those things about biology. They're the things that are living, so germs, diseases, blood, and bodily fluids. Chemical hazards are things that might be chemicals, so they're corrosive or toxic, or like an acid, they might eat through your pants. They might be hazardous and actually cause you to be really, really sick. And we might be talking about waste management. How do we actually get rid of this stuff when we're no longer using it? Physical hazards are how things could fall on you or poke you or jab you and make you hurt that way. So generally, sharp instruments, heavy objects, falling hazards are the main ones. Even broken glass is a physical hazard. There are often many hazards, and all of them should be recorded in the report. Your teacher will often highlight these hazards when you're ready to get started, so pay attention to what they're saying. If they say put on a lab coat, there's probably a good reason for that, and it should probably go in your safety. Here's a sample of safety. So in this experiment, students were asked to use a scalpel to cut open a heart. The scalpel is sharp and could be dropped on a foot or cut someone if it's not carried correctly. We know from reading the previous slide that this is a physical hazard, as the risk is the sharp object. So as a student, I'd need to say, physical hazard. Sharp scalpels could be dropped on feet or cut someone if not carried correctly. Always wear leather school shoes to protect your feet and walk with the scalpel pointing down to the ground. Always make sure the scalpel is placed safely in the dissecting tray when not in use, and always cut away from yourself. There's a pattern to what I'm saying here. What we've done is we've said the type of hazard. So is it biological, chemical, or physical? What the danger is, so how you could be hurt, and the precaution, how to keep you safe. And this is really handy because once we know how we could be hurt, it's easy to work out how we keep safe. Okay, remember you can pause again if you need to. Materials. This is the next section. What I need you to think about this time is, what did you use? Make sure you include everything and how many of each material you needed. So if you need 2 cotton buds, make sure you write 2 cotton buds, not just cotton buds. How big or how much do you need to use? And make sure you list them as dot points. So in this section, you need to make sure they're dot points in a list, so a column running down the page. Here's an example of a materials listed with a quantity, 1, and then the size, 3 centimetres by 4 centimetres. And what is it? It's a piece of paper. The method. Now, the method is a bit tricky. So when we write scientifically, we remove as much of the personal detail as we can. We don't actually want to talk about ourselves or what we're doing. We want to talk about what we're seeing or what we're doing. Okay, it isn't biased towards a particular outcome. We're not actually trying to tell the person what we think is going to happen. Writing scientifically is universal. It's the same all around the world. So it isn't important where you are, you'll understand the results. And that's really what we've been doing the entire time, but the method's a particularly tricky language to get practiced on. So make sure you're pausing this and come back to it as you write your prac report. So how do we write a method? There are 3 parts to the instructions. So when you're given a set of instructions, look for what the action is, so what was done. Look for what the object is that the action's affecting, so what was it done to? And then look for where the object is or how it should be done. That where or how it was done, we would like to have you underline in blue, and what it was done to, we're going to underline in red, and what was done, we're going to underline in green. And by using these colours, we can work out what parts of our sentence we actually need to rearrange. It's actually a really nifty pattern to this. So let's try them. Let's find them in the first instruction. Put gloves, safety goggles, and lab apron on. So using the three coloured pattern, we first of all want to underline the doing word in the sentence, and we want to underline it in green. So ask yourself, what was done? Well, in this sentence, it was put. Put is our doing word. Next thing, what was it done to? Well, it was done to the gloves, what was put on? What was put? The gloves, the safety goggles, and lab apron were put. So we have to underline them red because they're the object in this sentence. And it's a rather long object in this sentence. Sometimes it's just one thing. The next thing we need to highlight or underline in blue is how or where it was done. Now, if it's really important or really dangerous, you'll often find the blue bit at the start. But in other sentences, you'll find it just at the end here. So, how was it done? Well, where did we put the gloves, the safety goggles, and the lab apron? Well, we put them on. So here, when we say on, we're actually implying that we put them on ourselves. We don't have to say ourselves because we don't worry about the people. So the first point should look like green, red, and blue. What tense is this? Well, it's actually an instructional text and it's telling you to do it. So it's not past tense, it's more like present tense. So what is the quickest way to make it a past tense sentence? Because that's what we need to do when we write the method as a recount. Well, the fastest way is to say we put the gloves, safety goggles, and lab apron on. I want you to stop and think, what colour is we? And do we talk about the people or the science? Well, we don't talk about the people, so we need to change this sentence again. In science, we only talk about the science, focusing on the object or the action. So when we get we or she or he or they in a sentence, we actually need to try and remove it. And we do that by talking about what we did it to. So what happens if we change the order of the colours to make the object the focus? Well, we've tried putting the blue out the front, but does it make sense and does it change the tense? On we put gloves, safety goggles, and lab apron. Does that sound awkward? It does, doesn't it? So it can't be quite right yet. Let's try the reds at the front. Gloves, safety goggles, and lab apron we put on. Reads a bit better, but I think we need to change we for another past tense linking word to make this sentence make sense. If we change we for were or was, then it makes more sense. Was we use when there's only one thing, and were we use when there's more than one thing, a plural. So in this sentence we have three things: gloves, safety goggles, and lab apron, so we need to use were. Gloves, safety goggles, and lab apron were put on. Yep, that makes much more sense. Okay, so those past tense linking words, was or were, yep, they're in there too. Okay, so we've gone through that method checklist. Let's go through it again for when you're writing your next method. First of all, underline with the sentence, the instructions, with the three colours. So ask yourself, what was done? Colour that green. What was it done to? Colour that red. And where or how was it done? Colour that blue. Write what you did. Don't write what someone needs to do, okay? So it's always what you did, it's a recount. Make sure you move the reds together to the front of the sentence. Change the we to the past tense link word, were or was, and then you should have a pattern. Your pattern should be red, what was it done to? Was, were, what was it done to? What was done, and then how or where was it done? The conditions. The results. The results are a little bit tricky. There's more than one way to collect results, but it's really just saying after each step of the experiment, what did it look like, or what was the number, the value that we measured. So they can either be measurements, observations, or diagrams, and they're usually recorded in a table or just in a results section of your practical report as say scientific diagrams. So if you've got measurements, see if you can put them in a table. You'll usually have time and then something like centimetres or kilograms, or if you're studying force, it might be newtons. Okay, make sure you're as accurate as possible if you're using measurements. Observations require lots of adjectives. Think about painting a picture with words. What did it really look like? Does my red look the same as someone else's red? So if I just write red, is that enough? What did it feel like? Was there a temperature? Was there a sound? Was there a smell? Okay. Make sure you're using all of your senses when you're writing observations, and make sure you include as many adjectives as possible so that the person reading the results sees the same picture in their mind as you do. Diagrams are another very interesting way of collecting results, particularly when we're using microscopes or doing dissections. I'm going to run through again how you do scientific diagrams, but just remember they should be about 5 by 5 centimetres in size at the smallest. Sometimes they'll take up a whole A4 page. They should always be drawn in grey lead pencil, and they don't have shading. They are an outline only. Labels are used to show the key features and could be connected to the image with a straight line, just like our picture of the kidney over here. The only problem with this kidney is that you can see that this vein is coloured in grey to show that it's actually blue, and you can see that these patches here have also been shaded grey to show that they're a different section. So this person who wrote this diagram made it very easy for us to understand, but it's not actually a scientific diagram properly. So scientific diagrams, how do we do them? Well, they're an outline. So if I'm going to draw an animal cell, I would start by drawing just the outline of what I see. So here is what I see under the microscope. And I've just drawn one of them, as much detail as I possibly can. So this outside section, I know from my theory, is called the cell membrane. Sorry, just bear with me for a second while I label it. So you can see I've used a straight line to connect my words, and then I've written it as neatly as I can off to the side. So the cell membrane, being the gatekeeper of my cell, is now labelled on because I can see that on the microscope. I can see where the edge of the cell is. Now in this cell it was stained, and I could actually see this circle shape in the middle, and that is the nucleus. So again, as straight a line as possible. If I was doing this on paper, I'd actually use a ruler, and I'm going to label it the nucleus. Okay. And then there was all this stuff inside. So again, I'm going to rule a line as best I can, and I'm going to write the words cytoplasm, because that's what it is. I remember reading that in my school book. So here I go, cytoplasm. Can you see how the words aren't following the line? They're actually being written exactly horizontally. Well, as close as I can get on this computer. So make sure you're doing the same. Nice straight line to where you want to write the word, and then write the word horizontally. Make sure they're nice and clear. Now there's something missing from this cell, and that's a title. So I'm going to write that on, animal cell. And to show that that's a title, I'm going to underline it. Now I should probably say at what magnification I saw this at as well. So I'm going to write times 40, because that was the objective I was using at the time. Okay, so that was a basic scientific diagram. Now the discussion. There are 3 parts to a discussion: the analysis of results, the improvement and errors, and an optional one, group work and collaboration. Your teacher will tell you if you need to include that as well. In part 1 of the discussion, We're using our textbook, our work notes, and our results from the experiment to write about our aim. We're trying to remember what that purpose of the experiment was and now reflect back, well, what do my results tell me about that? So if my aim was to measure the effect that light has on plant growth, I'd probably be comparing how much more growth there was in the plant in the sunlight compared to the plant that was in the dark. Okay, I'd now explain why that result would have been like that. So not just, okay, it's bigger, but why would it have been bigger? What do I know about this topic that will help me answer that question? And I need to give as much detail as I can. Sometimes a graph can help me show trends or patterns in my measurement. And we'll do a different tutorial on how to draw a really good graph later. But make sure you're writing in factual paragraphs. Remember, topic sentence, evidence, summary. Not just question 1, question 2, question 3. In part 2 of the discussion, you're talking about the errors and the improvements. So you're saying, what's the possible error and how can the method be changed to reduce this error? So we're reflecting on what we did and how we could make it better next time. For example, if the paper was measured with a ruler but different rulers were used, there may be an error in the measurements. So the student might write, measuring the paper with different rulers can give different measurements. Always make sure the same ruler is used for each measurement. You can see from my colours here that we have pink and then purple. And down here in my example, I've also got pink and then purple. So you can see that it was what was the possible error and then how can the method be changed to reduce the error. Okay, part 3 of this discussion is that group work and collaboration. This is a reflection on how the roles that you used in your group helped you complete the experiment in the given time. Now, sometimes groups work really well and sometimes groups don't. So sometimes you want to say that there's something you'd like to change, and that's okay. Just explain what you would change and what the effect would have on the group. If it worked really well, say why it worked really well. And if you were doing this experiment again, would you take the same roles on? Well, if you say yes, tell me why. Conclusion. We're at the end. Yay! Summarise the information that you've discovered about the topic. You're going to go back to some paragraphs. About 2 this time. We're going to say, again, topic sentence, evidence and summary. We're going to do a factual recount. So again, we're saying what had happened, but we're making sure we're using facts and we're drawing on the evidence we've collected in our experiment to back ourselves up. So we're basically saying, what have we learnt from this? But we don't want to talk about emotions. We don't want to say we had fun or we like this one. We would like to avoid saying, I learnt. Instead of saying, I learnt, just tell me what you learnt. Sometimes when we're summarising the information, we've got new questions. So if you've got a new question that you'd like to know more about, say it. Say, this experiment opened my eyes up and made me wonder what would happen if the plant was in partial shade. A new question, new experiment. And that's how good scientists get their next research grant. Well, remember to re-watch and make sure you pause when you need to.